Hi, everybody. Very excited to be here to introduce our next-gen AI reasoning platform that allows us to generate on-demand, in silico, mechanistic whole-cell models. So it's not machine learning, because machine learning gives you correlation, association. Here it's about causation. And we want to apply that to develop what we call causation-based precision medicine. So rapidly about the team and myself, I have an MD, PhD in AI. I work in the field for 30 years. My first company, my first AI company, has been very successful. We were acquired for $630+ million, but it was mainly in finance. And at that time, life science is not the way it is today. So this second AI company is really focusing on life science. I have an amazing team that is multidisciplinary, from AI, biopharma, medicine. And I would like to honor Steve Howe, our dear friend, that just passed away last week after battling cancer. I'm sure a number of you know him. He's an amazing person, and we will consider him as part of the team forever. So the omics revolution, very important because it brings a new dimension to precision medicine. And data give you the what. In this case, genomics, epigenomics, and so on. But the real challenge is to interpret this data, so to get the how. So then you can understand what's going on. So we all know that the genome is very important for the cell. But what really matters is the gene expression, where you can really see the color of the cell, if I may say, its phenotype. So the thing is, you have a lot of technology, including machine learning and so on, to do association, statistical association between the phenotype, the genotype, and the phenotype. But we need to go deeper inside of the cell at the mechanistic molecular level. So you have thousands and thousands and thousands of scientists using different technology for that. And they are producing data, they process data for the only one thing, knowledge. In this case, the knowledge is about biochemical reactions, structure of the cell, etc. But unfortunately, this knowledge is so important today that nobody can acquire all the knowledge. The result, we have a fragmented knowledge acquisition. Worst, based on Miller, we can only process 5 to 9 concepts at the same time, which means that we underuse dramatically what we know and even more what is available. But even though with this very imperfect situation, we are successful to develop antibiotic, vaccine, immunotherapies, right? So what if we can unify all this knowledge together in one technology and let the scientists have access to it? This is what TNL we are doing. We have the technology to unify this knowledge into a reasoning network that has the power to connect all the dots. So this allows us to really approach what we call causation-based precision medicine, because we can understand the cause and effect. Now let's see what is the impact of this approach for drug R&D, diagnostic, and therapeutics. But before, I want you to illustrate more what really about reasoning compared to pattern recognition with machine learning, for example. So this is a simple puzzle. You have a lady and the tiger, and you have 2 rooms. And you have to find where is the lady, room 1 or room 2. For that, you have some knowledge. If the lady is room 1, then the sign 1 is true. If the tiger is room 1, then the sign 1 is false, etc. So if you spend 30 seconds, 1 minute, you will be able to infer that. How you do it? You start with an assumption, right? Let's suppose she's in room 1. And from there, you're going to infer a number of things until you get to a contradiction, a conflict with your knowledge. Then you challenge your assumption, and you do it again. So this is how we solve problems, and this is how TNL reasoning engine is doing it. So once you solve it, you will see that the lady is room 2 and the tiger is room 1. This illustrates at least 2 things. One is you can infer new knowledge from existing knowledge, and it has nothing to do with data. Now, maybe it takes you 30 seconds or 1 minute to do this one, but 9 rooms is going to take you hours or days, right? And the reason I present that is Now, if you replace lady, tiger, room by transcription factors, enzyme reactions, and so on, the cell has thousands and thousands and thousands of room in the lady and tiger, at least at the logic level, and then you have the quantitative level of it. So what we do with that? With this technology, we can generate on-demand, in silico, whole cell disease model for patients. How do we do it? You have the patient, and as you know, most of the disease translate that into the cell state. So we have the patient's cellular profile, and omics data allow us to access to it. We inject this omics data into our reasoning network, and we can produce, in a matter of 2 hours, any human cell type or state. Now, we have in silico patient cellular profile. So you have in silico model of the disease at the cellular level to play with for the drug diagnostic and so on. So the data give you the state of what's going on. The reasoning give you the dynamics. And only through the dynamics you can understand the causality. So let's go deeper to understand how it works. Each patient or group of patients will have 3 types of model: healthy, sick, and altered. The altered one is the sick data that we altered with a simulation of the drug by inhibiting some protein, for example. So if you compare the healthy with the sick, then you will understand the pathobiology of it, which allow you to have a more accurate target discovery, biomarkers discovery, and this, of course, enhance your patient stratification, diagnostic, and therapeutics. If you compare now the sick with the altered, then you will understand the impact of the drug, allowing you to do target validation, target ranking if you have already a list of candidates, repurposing, or combination. And lastly, you can optimize the clinical trial in silico. You run it in silico. So we do not claim that our model is perfect and complete, but we claim they are very useful. And this is how. So in target discovery, we were working on ulcerative colitis with a big pharma. We were able to find a druggable target, and it's druggable because it's already in phase 1, phase 2 for cancer. How we did it? From a publication where you have 18 patients, 12 healthy individuals with 51 cell type, in about 1 month, when we have all the data in the right format and so on, we were able to do this identification. From the publication, we extracted the transcriptomic data. And as you know, any data is noisy. Omics is even worse. In this case, among the 20 very well-known inflammatory markers for the macrophage, in this case, 12 of them were lower in the inflammatory data compared to the healthy one, which means if you use a pure data-driven approach, you already start with the wrong foot. So with the reasoning and the knowledge, the system were able to correct it because it takes into account the combination of the gene expression. So quickly, the gene 3 and the gene 2000 and so on are increased, then no way that range can be lower. Then we were focusing on the anterior side, having the healthy and the inflamed data. We put it into the system, we run it, so now we have the 2 models. We compare the 2 models, so now we can know what are the proteins that increase or decrease and why. Then we compare that with the macrophage. We can do that with the 50 other cell type to better understand what is specific to the anterior side. We got the 20 protein that has a very strong signal, and we focus on the X protein that has a high level because it's druggable. From there, we can do target validation because we injected that into our inflamed data to have the altered model. We do again the process to have now the 2 models. We can compare them and see what is the impact on the inflammatory markers in this case. Then you can use the model again to optimize the clinical trial. We run that in the group for each of the patients to see which one respond based on the biomarkers. Now we have the 2 groups. We can model these 2 groups, so we have the model of the responder, but at the same time, we can understand why they respond or not. Then with the same model, now we can run the in silico clinical trial. As you know, the in vivo testing is the most expensive part of the drug R&D. So here, before you go into this lengthy and costly thing, run in silico, giving you more insight about how to include or not the patient. Then, as another example, with this model, well, you can do drug repurposing. You can have hundreds, thousands of drugs and just run it. So then you can see, for example, in this case, drug 6 look interesting. Maybe we should focus on it, and we can understand why. So we are looking for 4 types of collaborations. Either you already have a disease focus, or you already have target candidate, or you just have a population that you want to understand what's going on, or you are already at the clinical trial stage. If you are at the first stage, then yeah, we can help you through the causation network to have target discovery, biomarkers discovery, repurposing, and combination. If you have already target candidate, then we can help you to do the validation and to rank them. So you can prioritize among, maybe you have 50 of them, which one you should start first, to have biomarkers discovery, in silico testing, and to have a better understanding of the mechanism of action. And of course, the clinical trial, as I already showed to you how we do it. So, and the population, you have to do 1 and 2. So, where we want to go from there. Today, this is how we solve problem, right? Each time you go to a biological conference, you have this kind of graphics. Because we cannot handle more than that. And there are thousands and thousands and thousands of these pathways. But we all know that this pathway has to interact to each other inside of the cell, and then between the cell, and then with the microbiome. This is way beyond human cognitive capacity today. TNL, we are already at the single cell level. We are looking for very strong partners and big funding to go to the multi-cell and the microbiome. And I would finish with this slide. It's the quote from Vijay Pandey, general partner, head of life science investment, Andreessen Horowitz. I'm sure you know the firm. They just raised $9 billion for their fund. So for him, today the industrialization of biology is happening, and it's enabled by AI. And this should lead to $1 trillion company. But AI is too dumb now. And the reason why he considered that AI is too dumb now is because it doesn't embed expertise. And expertise has 2 components, knowledge and reasoning. This is what TNL we are bringing on the table.